consequences of thyroid imbalance is so severe, I think it's worth revising or reviewing the functions of thyroid hormones. Here, definitely, at least at end of this session, okay, you would have studied in the first year. In case you have forgotten, at least end of the session, you should be able to outline the steps of synthesis of hormones. It will be useful in better understanding of mechanism of action of several anti-thyroid drugs. Okay, you can correlate that way. Similarly, hormones of receptors of thyroid hormones, how interaction takes place, and again for it will facilitate your receptor associated diseases maybe. Further, how the thyroid associated hormones are maintained in the blood. It would be TRF, it could be TSH or thyroid hormones. And understanding this axis, again it's useful for categorizing your thyroid disorders, whether primary or secondary. You can diagnose the by understanding the theoretical aspect of regulation of thyroid hormone. And finally, the key part of the topic is the functions of thyroid hormone. Definitely by understanding its influence, the metabolic or its role in growth and differentiation or the individual systems, you should be able to better explain the physiological basis for the symptoms associated with hypo and hyperthyroidism. In fact, the research on, I think in the field of physiology, pathology or medicine contributed a lot understanding the functions or actions of thyroid hormone. These are certain different milestones. Maybe it's a early findings, early contribution for the field of thyroid physiology or overall thyroid medicine. So as early as possible, there is a first report from the Clinical Society of London where they correlated hypothyroidism in adults, children with the destruction of thyroid gland. Similarly, after a few years, surgery or it's a surgical or a pathological procedure that is Emil Cocker later, I think pioneer, actually that is the pioneer study on thyroid gland for which he was awarded Nobel Prize also. Similarly, over the years, that is the receptor, the nuclear receptor was demonstrated, and the thyroid hormones, two different metabolic category, that is T3 and T4 were extracted initially from the sheep, and its usefulness in diagnostic usefulness also demonstrated. And one by one the physiological actions also documented its influence on metabolic or oxygen consumption or the critical for growth were quite well established. As hormones are metabolically active hormones are T3 and T4, right? So hormone secreted around 93% of metabolically active is the tetraiodothyronine, whereas the remaining 7% is triiodothyronine. Whereas metabolically inactive hormone is also secreted reversed T3, and the parafollicular cells secrete calcitonin. Here, the definitely normal concentration and deviation will be taught in detail by Professor Dr. Sampath, that is especially the deviation from the normal value, right? Because this table clearly mentions hormone is transported as a free as well as bound form, that is thyroid binding globulin. Okay, so clinically it's quite useful index assessing TSH or free or bound form of thyroid hormones. I think it's worth appreciating the differences between T3 and T4. Functionally, they have similar whereas what we can say is qualitatively they are similar, whereas the rapidity and intensity of T3 is almost 4 times greater than T4. So previous slide you have seen T4 is abundant, right? T3 is a small fraction, whereas T3 is more potent than T4. Further, the all circulating T4 has to convert into T3 before it binds to the receptor. In the target cell, T4 is converted into T3, or receptor is sensitive mainly for T3. So half-life is shorter and concentration is less, however more potent than T4. The T3 is more potent than T4. Follicular cell definitely higher concentration of iodine, around that is, it shows around, in the normal cell around 30 to 50 times greater than the ECF. In spite of that, the active transport of iodine takes place through the basement membrane, because it's a secondary active transport, it's sodium iodine symport, and the mechanism is facilitated by TSH, thyroid stimulating hormone. And here again, as applied aspect, the cyanides in fact prevents the synthesis of thyroid hormone by blocking this particular step. The iodine trapping mechanism is blocked by thiocyanate, one of the anti-thyroid drug. So you have now iodide is within the follicular cell, the first step. Second, of course, the synthesis of thyroglobulin. It's a continuous protein synthesis takes place in the colloid. Thyroglobulin synthesis consists of tyrosine molecule. You know the precursors are basically iodine and tyrosine, right? So tyrosine is in the form of thyroglobulin. Once again, with the influence of THS, that is synthesis of thyroglobulin takes place. Further, the next step, that is iodine can combine, what we call is iodination of thyroglobulin. To iodination of thyroglobulin, there should be oxidation of iodide. Okay, iodide in the form has to convert into nascent iodine or molecular iodine. So iodide is converted into iodine in presence of thyroid peroxidase. Okay, enzyme thyroid peroxidase as well as TSH. So any agent which blocks thyroid peroxidase once again prevents the synthesis of hormone T3, T4. So one of the, that is thiouracils, well-known anti-thyroid drug, which prevents the conversion of the iodide into iodine as well as further organification of the iodine, the fourth step, that also prevented by thiouracil. The final step is, that is formation of T4 and T3 by coupling reaction. So initially one iodine binds with tyrosine, you call it as monoiodotyrosine, right? When two iodine with tyrosine, it's diiodotyrosine. So final step is coupling of two molecules of diiodo forms T4, right? One monoiodo forms T3. So I think you can recollect this. We studied the first year as step iodine trapping, right? Synthesis of thyroglobulin, oxidation, organification and coupling. So here the whole process is greatly influenced by thyroid stimulating hormone. In the absence of this hormone, very less amount of T3, T4 synthesized. Meanwhile, the growth and vascularity also greatly suppressed. That's why thyroid disorders can be hypothalamus origin, it can be pituitary origin, or just the primary thyroid gland origin. Anyway, it's missing. Short loop is regulation through TSH. Hypothalamus is suppressed, right? Whenever regulation is by TRH, it's called ultra short loop regulation. So overall, HPT axis is essential to maintain normal T3, T4 level. Before explaining the functions, how exactly the operation take, how exactly the interaction of hormone with the receptor takes place. You would have studied there is a mechanism, entirely different mechanism for steroid. Peptide is different. This is which class of hormones? Amines, right? The third category, the amines. Here, this the molecular basis is very similar to steroid hormone action. Okay, that is, the receptors are present within the cell. We call it as intracellular receptor. For peptide, we have the surface receptors. So here the intracellular receptors, and the receptors not in the cytoplasm, unlike steroid hormones. They are present in the cytoplasm. Here it is within the nucleus. Intracellular, but they are the nuclear receptors. So the hormones generally transported through a carrier protein. It's transported through the carrier protein. Sorry, audio, I'll stop it. Okay, that is transported and lipophilic and easily diffusible through the membrane. So both T3 and T4 are diffuse into the cytoplasm, where in the cytoplasm most of the T3 is so T3 also diffuses quite slow. So T3, T4 diffuses. Conversion of T4 into T3 into T4 takes place. Then only it can diffuse to the nucleus, binding to nuclear receptor. Later, hormone, here it's a thyroid response element in the DNA, right? Hormone receptor complex, now its a receptor is quite activated once the binding of hormone. So hormone receptor complex binding leads to mRNA synthesis as well as mRNA synthesis, and finally translation for the new proteins. So you got it, I don't have to replay it, right? So mRNA, that is the transcription for new genes in the nucleus. Finally, these are mRNA diffuses back into cytoplasm, where translation for specific proteins. So wide variety of proteins are specific, wide variety of proteins are synthesized. They can be structural, they can be carrier protein, they can be enzymes. Finally, the thyroid mechanism of action is achieved. Either as an enzyme, it can accelerate any metabolic function. As a structural protein, it can promote the growth, right? As a carrier protein, it can fluctuate the transport. So one of those, through newly formed protein, finally, the physiological action can be achieved.